Hi, everyone, and thank you for the opportunity to talk today about our technology, which is a soft adhesive hydrogel, which can be applied to the surgical resection cavity of brain cancer patients to mediate therapeutic efficacy. And of course, while I'm the person up here talking today, this work wouldn't be possible without our team of scientists, engineers, clinicians, entrepreneurs, and people who are just generally passionate about helping patients. So why brain cancer? Well, despite all the medical innovations that we've had in the past 50 years, brain cancer therapy hasn't changed significantly. In fact, as you can see by the rate of death up here, with 90% of glioblastoma patients, which is the most aggressive form of brain cancer, they die within 2 months of diagnosis. For context, for all cancer types, 50% of patients will be alive 5 years after diagnosis. So why is this? Let's take a look at what is clinically used. So for glioblastoma patients, they actually go a pretty difficult and aggressive regimen. First, they'll undergo surgery, but you can appreciate that surgery might not be able to eliminate all tumor cells, as shown by this arrow. To get rid of these tumor cells, chemoradiotherapy is often used. However, it's not specific and may not be able to reach all tumor cells, so they start to recur. This results in local progression, as shown here, and finally, as infiltration of healthy brain structure. So why do these therapies fail? As you may have guessed, we're unable to get rid of these cells. But as one of our colleagues at Dana-Farber Cancer Institute elegantly put it, the top 3 reasons that brain cancer therapies fail can be summarized in 2 words: drug delivery. As a drug delivery scientist, I can appreciate this. Over decades of research have shown that most of drugs, over 98% that have been developed in the clinic, are unable to cross the blood-brain barrier. For those of you that aren't familiar, the blood-brain barrier is an anatomical structure that is specifically designed to allow vital nutrients, such as oxygen and glucose, to cross while keeping out pathogens, such as viruses and bacteria. However, the blood-brain barrier is unable to distinguish between healthy things like drugs crossing and things like bacteria and viruses, which prevent most therapies from reaching the brain tumor, accumulating in therapeutic doses, and mediating therapeutic efficacy. Our solution to this challenge is to use a local adhesive hydrogel that can mediate the controlled delivery of any type of therapeutic when sprayed or injected into the tumor resection cavity. It is promising because, unlike other hydrogels, it is soft but tough. It can withstand many types of surgical forces while continuing to be able to mediate tunable drug delivery of any type of therapy. This is important because it allows drugs, regardless of their ability to cross the blood-brain barrier, to be able to be applied directly to the tumor where they need to act. We have developed microscopic images of our hydrogel that you can see here on the left. You can see the polymeric matrix with a drug or model drug shown in red. You can also see that when we implant a droplet the size of a raindrop into the mouse brain, that can persist for up to 3 weeks. However, I'm sure the question you're all wondering about is, how efficacious is this? So to orient you, here you can see that mice that we have implanted with a brain tumor intracranially will die quickly without being treated. Next, you can see that with the current oral temozolomide chemotherapy regimen, that mice continue to die rapidly. Again, this is what is used clinically. Next, we tested whether or not simply bypassing the blood-brain barrier with local injection of a potent chemotherapy would result in improved survival, and we see that it does not. When we add in our hydrogel, however, you can see that this line increases quite significantly, and we have over 50% survival. This is a result of our controlled, mediated, sustained adhesive hydrogel being able to maintain drug at the site of interest. And finally, when combined with the current chemotherapy, oral temozolomide, we see an even further improvement. It is able to control the delivery of the drug and work in synergy with oral temozolomide. However, as I told you, tumors often recur. And to mimic this in mice, we actually re-challenge them with another tumor in the other hemisphere. This allows us to assess whether immune memory is formed, because the immune system is capable of fighting things regardless of its timing or location if it knows what to look for. And here we see that after challenging all of our mice, that we have 100% survival for every single mouse that was treated with our hydrogel therapy. And in fact, These mice have been alive for over a year now. So the question is, what comes next? Well, we're motivated to bring this therapy to patients because this is why we do what we do. Furthermore, we're always, as scientists, looking to increase the bar and seeing how we can do better. And to that end, we've recently tried a nanoparticle immunotherapy in our hydrogel and delivered it locally. And as you can see here, we've achieved 100% survival. So what do we need next to make this happen? Well, we're looking to build a venture and spin out at the end of this year. We're also looking to identify investors to help us rapidly accelerate to and through the clinic. And of course, as scientists, as I mentioned, we're always looking for great collaborations or partnerships to make sure that we are delivering the best possible therapy to patients. So to summarize, as I started, in 2 years, most brain cancer patients will succumb to their disease. Well, as most of you can appreciate, with the COVID-19 vaccines, we've seen how drug delivery systems, when paired with the right therapy, are able to shape the pace of a global pandemic. We expect to achieve the same transformative effect for brain cancer patients. With your help, we will be able to bring brain cancer patients and their families hope by delivering on this technology. Thank you.
Thank you.